Pre-IPO institutional research report Doudna's CRISPR spin-out bets on once-a-decade cholesterol control. NASDAQ, SCTX. Section 1. Executive Summary. Scribe Therapeutics Inc. is an Alameda, California-based clinical-stage biotechnology company developing next-generation CRISPR gene editing and gene silencing therapies. Co-founded in 2017 by Nobel Prize-winning CRISPR pioneer Dr. Jennifer Doudna, alongside CEO Dr. Benjamin Oakes, a former researcher in Doudna's own laboratory at UC Berkeley. As of this report's date, Scribe has not yet completed its IPO. The company filed an amended registration statement on July 20, 2026, setting terms of 7,150,000 shares at an expected price of $13 to $15 per share, and pricing is anticipated imminently, with NASDAQ trading under the ticker SCTX, expected to follow shortly thereafter. In plain terms, Scribe has engineered its own alternative to the well-known CRISPR-Cas9 gene editing system, based on a different bacterial enzyme called Cas9, which the company says is smaller and more precise. Its lead program, STX1150, doesn't permanently rewrite a patient's DNA the way classic gene editing does. Instead, it uses a technology called ELXR, which stands for Epigenetic Long-Term X Repressor, to durably switch off a specific gene called PCSK9, which is well known to drive high cholesterol, without altering the underlying genetic code. The pitch to investors and doctors alike: a single treatment that could keep LDL cholesterol low for years, or potentially a decade or more, versus the twice-yearly or twice-monthly injections patients currently need with drugs like Repatha or Leqvio. The company has already begun a first-in-human clinical trial of STX1150 in Australia under regulatory clearance from that country's Therapeutics Goods Administration, with initial data expected in the first half of 2027. Two additional pipeline programs, STX1200 and STX1400, target other genetic drivers of cardiovascular disease, specifically LPA and triglycerides respectively, and are expected to enter human trials in 2027 and 2028. Scribe has also formed strategic collaborations with Sanofi and Eli Lilly, both of which lend third-party validation to its underlying gene editing platform. Key facts at a glance. Core technology: engineered Cas9-based gene editing, XE, and epigenetic gene silencing, ELXR. Lead program, STX1150, epigenetic silencing of PCSK9 for durable LDL cholesterol reduction. Clinical status: Phase 1 first-in-human trial underway in Australia. Initial data expected first half of 2027. Strategic partners: Sanofi and Eli Lilly. Collaboration agreements: Sanofi is also participating in a concurrent private placement. Q1 2026 collaboration revenue, $2.2 million versus $17.1 million in Q1 2025. Q1 2026 net loss, $17.4 million versus $3.5 million in Q1 2025. Accumulated deficit, $175.1 million as of March 31, 2026. This report is intended to give both professional and retail investors a clear-eyed, plain English view of Scribe's science, its financial position ahead of its public listing, and the key risks and catalysts likely to matter over the next 12 to 24 months. It is not a recommendation to buy, sell, or hold SCTX shares. Section 2. Investment Thesis. The bull case for Scribe centers on the strength of its scientific pedigree and the size of the market it's targeting. Cardiovascular disease driven by high cholesterol affects a vastly larger patient population than the rare orphan diseases most first-generation CRISPR companies have historically pursued, and durable, infrequent dosing therapies are exactly what large pharmaceutical partners like Sanofi and Eli Lilly look for when validating a platform through collaboration deals. Co-founder Jennifer Doudna's Nobel Prize-winning status in the company's proprietary Cas9 enzyme, positioned as a genuinely differentiated alternative to the more widely used Cas9, gives Scribe real scientific credibility relative to many earlier-stage biotech IPO candidates. The bear case is equally straightforward. Scribe is testing an entirely novel mechanism, epigenetic gene silencing rather than classic gene editing, in humans for the very first time, with zero human efficacy or safety data available as of this report's date. The company's own numbers show collaboration revenue collapsing from $17.1 million to $2.2 million, and net losses more than quadrupling year over year in the most recent quarter, reflecting the lumpy, milestone-driven nature of partnership revenue rather than product sales. With only about $100 million expected from the IPO, Scribe will likely need substantially more capital to fund STX 1150 through later-stage trials and to advance STX 1200 and STX 1400, all before any of the company's 3 lead programs has generated a single data point in human patients. Thesis in one sentence. Scribe Therapeutics offers exposure to a scientifically credible, well-partnered, next-generation gene editing platform aimed at a massive cardiovascular market, but it remains in the most literal sense a preclinical proof-of-concept story until its first human trial data arrives in 2027. Section 3. Company Overview. Legal Entity. Scribe Therapeutics Inc., a Delaware corporation. Headquarters, 1150 Marina Village Parkway, Alameda, California. Exchange and ticker, NASDAQ Global Market, SCTX expected. Year founded 2017. Co-founder and CEO, Dr. Benjamin L. Oakes. Co-founder, Dr. Jennifer A. Doudna, 2020 Nobel Laureate in Chemistry for CRISPR-Cas9 gene editing. Other co-founders, Dr. Brett Stahl and Dr. David F. Savage. IPO filing status, Form S-1A, Amendment Number 2 filed July 20, 2026. Pricing not yet completed as of this report's date. Underwriters Leerink Partners, Goldman Sachs, Guggenheim Securities, and Wells Fargo Securities. Corporate milestones. 2017, company founded by Benjamin Oakes, Jennifer Doudna, Brett Stahl, and David Savage, spinning out engineered CRISPR technology developed in Doudna's UC Berkeley lab. 2021, completes oversubscribed $100 million Series B financing led by Avoro Ventures and Avoro Capital Advisors, joined by OrbiMed and others. 2025, forms strategic collaborations with Sanofi and Eli Lilly, presents preclinical STX 1150 data showing greater than 95% PCSK9 reduction in primary human hepatocytes in vitro at the American Heart Association's 2025 scientific sessions. January 2026, announces plans to enter the clinic with STX 1150 at the 44th Annual J.P. Morgan Healthcare Conference. May 21, 2026, achieves regulatory clearance in Australia, TGA, to initiate first-in-human clinical study of STX 1150. June 19, 2026, awarded more than $25 million in non-dilutive grant funding from the California Institute for Regenerative Medicine, CIRM, to support its CRISPR-based gene editing programs. July 2, 2026, publicly files Form S-1 with the SEC for a proposed NASDAQ IPO. July 20, 2026, files Form S-1A Amendment Number 2, setting terms, 7,150,000 shares at $13 to $15 per share, announces concurrent Sanofi private placement of approximately $7.5 million. Approximately July 23 to 24, 2026, anticipated IPO pricing and NASDAQ trading debut under ticker SCTX. Section 4, Business Model. Scribe operates a classic clinical stage biotech business model, built on 2 parallel tracks. The first is wholly owned drug development. Scribe funds and runs its own clinical trials for its lead cardiometabolic programs, STX 1150, STX 1200, and STX 1400, with the intention of eventually either commercializing these therapies itself or partnering and licensing them once they've demonstrated meaningful clinical value. The second is platform collaboration. Scribe licenses access to its underlying Cas-X, XE gene editing, and ELXR gene silencing technology platform to large pharmaceutical partners, currently Sanofi and Eli Lilly, for their own drug development programs in other disease areas, in exchange for upfront payments, milestone payments, and potential future royalties. In plain terms, think of Scribe as running 2 businesses at once. In the first, it's a traditional drug developer betting its own capital on a specific cholesterol-lowering therapy reaching approval. In the second, it's a technology licensor, renting out its molecular tools to bigger pharmaceutical companies that want to build their own therapies using Scribe's gene editing platform, similar in spirit to how a semiconductor company might license its chip designs to multiple manufacturers. This collaboration revenue is Scribe's only current source of income, since none of its own wholly owned programs have reached a stage where they could generate product sales. How to think about it. Nearly all of Scribe's revenue today comes from pharma partners paying to use its gene editing toolkit for their own research, not from selling any therapy of its own. The company's stock market value depends almost entirely on the market's belief that its own STX 1150 program will eventually prove safe and effective in the clinic. Section 5. Products and Services. CRISPR by Design Platform. Rather than using the widely known Cas9 enzyme, the original CRISPR gene editing tool, co-discovered by Doudna herself, Scribe has engineered a different bacterial enzyme called Cas9 into its proprietary XE technology, which stands for X editing. The company says Cas9 is naturally smaller than Cas9, which can make it easier to package and deliver into cells, and that its engineered versions offer improved editing efficiency, specificity, and a broader therapeutic window, potentially reducing the risk of unwanted off-target genetic changes. ELXR, epigenetic silencing without editing DNA. Scribe's lead program does not use XE to cut and edit DNA at all. Instead, it uses a related but distinct technology called ELXR, epigenetic long-term X repressor, which attaches to a target gene and adds chemical off-switches, specifically methyl groups, that suppress the gene's activity without permanently altering the underlying DNA sequence. Because nothing is physically cut or rewritten, the company argues this approach may carry a different and potentially more favorable safety profile than permanent gene editing, while still delivering years-long, and management hopes decade-long durability from a single treatment. Pipeline. STX 1150 targets PCSK9 for LDL cholesterol reduction. Mechanism. ELXR epigenetic silencing. Status. Phase 1 first-in-human trial underway in Australia. Data expected first half of 2027. STX 1200 targets LPA or lipoprotein A. Mechanism. XE gene editing. Status. Preclinical. Phase 1 anticipated 2027, supported in part by CIRM grant funding. STX 1400 targets triglycerides. Mechanism. XE gene editing. Status. Preclinical. Phase 1 anticipated 2028, supported in part by CIRM grant funding. All three lead programs target genetically and clinically validated drivers of atherosclerotic cardiovascular disease, meaning the underlying biology is already well established in medicine. Scribe's bet is not on discovering new biology, but on using its gene editing and gene silencing tools to deliver a durable, potentially one-time treatment where today's standard of care requires ongoing injections. Section 6. Industry Overview. Gene editing and gene silencing sit within the broader genetic medicine industry, which has evolved considerably since the first CRISPR-based therapy, Casgevy, from Vertex Pharmaceuticals and CRISPR Therapeutics, won FDA approval for sickle cell disease and beta thalassemia in late 2023. Early gene editing approvals focused almost exclusively on rare, single-gene orphan diseases affecting small patient populations, where the enormous cost and complexity of gene therapy could be justified by the severity of the disease and the absence of alternative treatments. Scribe is part of a newer wave of companies attempting to extend gene editing and silencing into much larger, common disease populations, in this case, the tens of millions of people worldwide with elevated cardiovascular risk from high LDL cholesterol, lipoprotein A, or triglycerides. This is a meaningfully different commercial opportunity than rare disease gene therapy. The addressable patient population is vastly larger, but so is the bar for safety, given that these patients could otherwise be treated for years with well-tolerated, already approved injectable medicines. The broader gene editing biotech sector has struggled with public market enthusiasm since its 2021 peak, and the sector's IPO window has been largely shut for gene editing specific companies since 2024, when San Francisco Bay Area biotech Metagenomi raised just under $94 million in the last gene editing IPO prior to Scribe's. If successful, Scribe would become the 14th venture-backed biotech to price an IPO in 2026. In a year that has already produced two of the largest biotech offerings ever recorded, suggesting the broader biotech IPO market, if not the gene editing subsector specifically, has shown renewed strength. Section 7. Market Opportunity. Atherosclerotic cardiovascular disease remains the leading cause of death globally, and elevated LDL cholesterol is among its most well-established, treatable risk factors. Millions of patients already take PCSK9 targeting medicines like Amgen's Repatha or Novartis's siRNA-based Leqvio, and Scribe's central pitch is that its epigenetic silencing approach, could eventually offer meaningfully longer-lasting cholesterol reduction from far less frequent dosing, potentially a single treatment providing years of benefit versus the twice-yearly or more frequent dosing regimens of today's therapies. Beyond STX 1150's specific PCSK9 target, Scribe's STX 1200, targeting LPA, a genetic cardiovascular risk factor with no currently approved targeted therapy, and STX 1400, targeting triglycerides, extend the same durable silencing thesis to additional large, underserved cardiovascular risk markets. If Scribe's platform proves broadly applicable across these three programs, the company would have a multi-target pipeline addressing overlapping but distinct patient populations within cardiometabolic disease, a considerably larger and more commercially attractive market than the rare disease indications most earlier CRISPR companies have pursued. How to think about the opportunity. The clinical and commercial opportunity here is genuinely large if the science works, but if the science works is the entire question. STX 1150 has only just begun dosing its first human patients, and neither STX 1200 nor STX 1400 has entered the clinic at all as of this report's date. Section 8. Competitive Landscape. CRISPR Therapeutics, traded on NASDAQ under CRSP, co-developer of Casgevy, the first approved CRISPR therapy, more clinically advanced than Scribe, but focused on different, largely rare disease indications. Intellia Therapeutics, traded on NASDAQ under NTLA, in vivo CRISPR Cas9 editing collaborating with Regeneron on multiple late-stage programs, a frequently cited direct platform competitor. Beam Therapeutics, traded on NASDAQ under Beam, base editing a more precise variant of gene editing across several disease areas. Verve Therapeutics, traded on NASDAQ under Verve, base editing specifically targeting PCSK9 and ANGPTL3 for cardiovascular disease, arguably Scribe's most direct clinical stage competitor, given the overlapping PCSK9 target and cardiovascular focus. Prime Medicine, traded on NASDAQ under PRME, prime editing approach, a further refined gene editing technique, broader disease focus. Editas Medicine, traded on NASDAQ under EDIT, earlier CRISPR-Cas9 pioneer, largely refocused on in vivo editing following past clinical setbacks. Established PCSK9 therapies, Amgen's Repatha, Novartis's Leqvio, and Sanofi Regeneron's Praluent are already approved, widely prescribed injectable cholesterol-lowering drugs that represent both the standard of care Scribe hopes to disrupt and the practical benchmark its therapy must beat on convenience, safety, and durability. Where Scribe stands. Scribe's clearest differentiation is mechanistic, while Verve Therapeutics pursues permanent base editing of PCSK9, and RNAi-based drugs like Leqvio require repeat dosing every 6 months. Scribe's ELXR epigenetic silencing approach aims to sit in between, offering multi-year or longer durability without permanently altering a patient's DNA. Whether this middle ground positioning translates into a superior safety and efficacy profile in actual human trials remains entirely unproven, and Scribe is meaningfully earlier in clinical development than several of the public competitors listed above, most of which already have some human safety and/or efficacy data in hand. Section 9. Management Team. Dr. Benjamin L. Oakes, co-founder, president, and chief executive officer, holds a Ph.D. in biochemistry and molecular biology from the University of California, Berkeley, and a B.A. from Colby College. Joined Jennifer Doudna's lab at UC Berkeley to help engineer new CRISPR molecules before starting his own lab and eventually co-founding Scribe. Has publicly described his ambition to move gene editing beyond rare diseases into common conditions like high cholesterol, stating he views the company's ELXR technology as a potential end state for RNA interference-style therapies, requiring, in his own words, perhaps only a booster dose once every decade or two compared to the twice-yearly dosing of existing drugs like Leqvio. Dr. Jennifer A. Doudna, co-founder, recipient of the 2020 Nobel Prize in Chemistry for the co-discovery of CRISPR-Cas9 gene editing, and one of the most recognized scientists in the field of molecular biology. A professor at UC Berkeley, Doudna is also a prominent public voice on the ethical implications of genome editing. Her scientific reputation lends Scribe substantial credibility with both the scientific community and prospective pharmaceutical partners. Dr. Brett Stahl and Dr. David F. Savage, co-founders, both scientific co-founders alongside Oakes and Doudna, contributing to the original development of Scribe's engineered Cas-X and CRISPR by design platform technology. Finance leadership David Parrott joined Scribe as Chief Financial Officer, arriving from Barclays, where he worked prior to joining the company, bringing capital markets and banking experience relevant to Scribe's transition into a public company. Section 10, Financial Performance Key Metrics by Year. FY 2024, Collaboration Revenue, $27.4 million, Net Loss, $47.8 million. FY 2025, Collaboration Revenue, $51.2 million, Net Loss, $21.8 million. In plain terms, Scribe's only revenue comes from its pharma collaboration deals with Sanofi and Eli Lilly, not from product sales, since none of its own drug candidates has reached the market. This revenue is inherently lumpy. It depends on the timing of upfront payments, research milestones, and accounting recognition rules tied to specific collaboration agreements, rather than steady, predictable demand. That lumpiness is clearly visible in the year-over-year swing between 2024 and 2025, and even more dramatically at the quarterly level. Capital raising history. Scribe has historically been funded through venture capital, including a $100 million Series B financing completed in 2021, led by Avoro Ventures and Avoro Capital Advisors, joined by OrbiMed and other institutional investors. The company's broader venture investor base includes Andreessen Horowitz. Both Sanofi and Eli Lilly have also provided capital and validation through collaboration agreements, and Sanofi is participating in a concurrent private placement alongside the IPO of approximately $7.5 million. Accumulated deficit. As of March 31, 2026, Scribe's accumulated deficit, the cumulative total of all losses since the company's founding, stood at $175.1 million, reflecting nearly a decade of R&D investment in a platform that has not yet generated product revenue. Section 11. Detailed Analysis. Latest Quarterly Financials Q1 2026. Revenue. Collaboration revenue was $2.2 million in Q1 2026, down sharply from $17.1 million in the year-ago quarter. This is not evidence of a deteriorating business. It reflects the inherently uneven timing of milestone and upfront payments under Scribe's collaboration agreements with Sanofi and Eli Lilly, which can cause revenue to swing significantly from quarter to quarter, independent of the platform's underlying scientific progress. Net income. Net loss was $17.4 million in Q1 2026, compared to a $3.5 million net loss in Q1 2025, a more than fourfold increase. This widening reflects both the sharp decline in collaboration revenue in the quarter and continued increased spending to advance STX 1150 into and through its first-in-human clinical trial, along with preclinical work on STX 1200 and STX 1400. Guidance. As a pre-IPO clinical stage biotech, Scribe has not issued formal revenue or earnings guidance. Management's forward-looking commentary centers instead on clinical and regulatory milestones. Initial STX 1150 data expected in the first half of 2027, and anticipated Phase 1 trial starts for STX 1200 and STX 1400 in 2027 and 2028, respectively. Management commentary. CEO Benjamin Oakes has described the initiation of the STX 1150 clinical trial as a pivotal moment for Scribe, stating that years of iterative engineering to enhance the potency and specificity of our CRISPR technologies has culminated in our first clinical program designed to provide access to the effects of known cardioprotective genetics, potentially freeing patients from decades of treatment burden. Management has also emphasized the strategic significance of the Sanofi and Eli Lilly collaborations as external validation of the company's core gene editing platform, independent of STX 1150's own eventual clinical outcome. Key changes versus previous quarter. Collaboration revenue fell sharply quarter-over-quarter and year-over-year, reflecting the lumpy timing of partnership payments rather than any change in the underlying collaborations. Net loss more than quadrupled year-over-year, driven by both lower revenue recognition and continued increased R&D spend supporting the STX 1150 clinical trial launch. The company received TGA clearance and dosed its first patients in the STX 1150 first-in-human trial during this period, the single most important operational milestone in the company's history to date. Scribe also received more than $25 million in non-dilutive serum grant funding during this broader period, supporting continued preclinical work on STX 1200 and STX 1400 without additional dilution to shareholders. Section 12. Growth drivers, positive STX 1150 clinical data. Favorable safety and PCSK9 lowering efficacy data from the ongoing Australian first-in-human trial, expected in the first half of 2027, would be the single most important value-creating event realistically available to the company over the next 18 months. Pipeline expansion into the clinic. Successful advancement of STX 1200 targeting LPA and STX 1400 targeting triglycerides into Phase 1 trials in 2027 and 2028 would demonstrate the platform's applicability beyond a single program. Deepened or expanded pharma partnerships. Additional collaboration agreements, expanded scope with existing partners Sanofi and Eli Lilly, or new upfront and milestone payments would both validate the platform and extend Scribe's cash runway with non-dilutive capital. Continued non-dilutive grant funding. Scribe's track record of securing CIRM grant funding, over $25 million awarded in June 2026 alone, demonstrates an ability to fund pipeline work without diluting shareholders as heavily as pure equity financing would require. Scientific credibility and conference visibility. Regular presentation of preclinical and clinical data at high-profile venues, including ASGCT, the European Atherosclerosis Society Congress, and the American Heart Association Scientific Sessions, helps build the scientific and investor credibility needed to support future financings and partnerships. Section 13. Key risks. No human efficacy or safety data yet. STX 1150 has only just begun first-in-human dosing, with initial data not expected until the first half of 2027. All claims about the therapy's potential durability and safety remain based on preclinical, in vitro, and animal data as of this report's date. Novel, unproven mechanism. Elixar's epigenetic silencing approach has never been validated in human patients for any indication. Even if PCSK9 suppression is achieved, the durability, reversibility, and long-term safety of the approach remain genuinely open scientific questions. Concentrated, overlapping pipeline. All three lead programs, STX 1150, STX 1200, and STX 1400, rely on the same underlying Cas-X, XE, and ELXR platform and target the same broad disease area of atherosclerotic cardiovascular disease. A platform-level safety signal or delivery technology setback could simultaneously impair multiple programs at once. Lumpy, unpredictable collaboration revenue. Revenue swung from $17.1 million to $2.2 million quarter over quarter, and net losses more than quadrupled, illustrating how dependent the company's near-term financials are on the timing of partner milestone payments rather than any steady operating trend. Modest IPO proceeds relative to capital needs. At roughly $100 million, the planned IPO raise is relatively small relative to the capital typically required to advance 3 separate gene therapy programs through Phase 1, Phase 2, and eventually Phase 3 trials. Substantial additional, potentially dilutive financing is likely to be required well before any program reaches commercialization. Ex-US regulatory pathway for the first trial. STX 1150's first-in-human trial is being conducted in Australia under TGA clearance, rather than under a US FDA investigational new drug application. The company will still need to separately engage the FDA to access the primary US commercial market, introducing additional regulatory timeline and outcome uncertainty. Intense, well-funded competition. Verve Therapeutics is pursuing a directly competing PCSK9 targeting gene editing approach already in clinical development, while Intellia, Beam, CRISPR Therapeutics, Prime Medicine, and Editas all compete for capital, talent, and pharma partnerships across the broader gene editing landscape. Already approved PCSK9 inhibitors, Repatha, Leqvio, and Praluent, also represent a well-tolerated, effective standard of care that any new therapy must clearly outperform on convenience or outcomes to gain adoption. Weak recent history for gene editing IPOs. The last dedicated gene editing company IPO, Metagenomi in 2024, raised under $94 million, and the subsector's broader public market reception has been muted since the 2021 biotech peak, suggesting investor appetite for early-stage gene editing stocks specifically remains an open question, even amid a stronger, broader biotech IPO market in 2026. IPO not yet priced. As of this report's date, Scribe's IPO has not yet priced. Final offering price, proceeds, and trading debut timing remain subject to change based on market demand and conditions. Section 14. Valuation. Traditional valuation metrics such as price to earnings or price to sales are not meaningful for Scribe, given its minimal, non-recurring collaboration revenue and lack of any approved product. As with other pre-commercial clinical stage biotechs, the market will effectively be pricing SCTX based on the perceived probability of STX 1150's eventual clinical and regulatory success, the value of the broader 3-program pipeline, and the ongoing strategic value of the Sanofi and Eli Lilly collaborations, discounted heavily for the years of clinical development risk that remain. Key metrics. Proposed price range, $13 to $15 per share. Shares offered, 7,150,000. Expected gross proceeds, approximately $93 million at the low end to approximately $107.2 million at the high end. Implied market valuation at the top of the range, approximately $242.7 million, based on shares outstanding disclosed in the S-1A. Concurrent private placement, approximately $7.5 million from Sanofi. At the top of its proposed range, Scribe would be valued at roughly $243 million, a relatively modest valuation for a company with a Nobel laureate co-founder, 2 major pharma collaborations, and a 3-program pipeline, but one that appropriately reflects the very early clinical stage of the lead asset and the muted recent public market reception for gene editing IPO specifically. Section 15. Upcoming catalysts: IPO pricing and NASDAQ trading debut, approximately July 23 to 24, 2026. The company's first real market test of investor demand for its story. Initial STX 1150 clinical data, first half of 2027, the single most important value-defining event for the company. Will provide the first human evidence of whether ELXR-based epigenetic silencing works as intended. STX 1200 Phase 1 trial initiation, targeted for 2027, would demonstrate the platform's applicability to a second cardiovascular target, LPA. STX 1400 Phase 1 trial initiation, targeted for 2028, would demonstrate the platform's applicability to a third cardiovascular target, triglycerides. First post-IPO quarterly report, date not publicly available. Will give investors their first standalone look at operating trends and cash runway as a newly public company. Potential new or expanded pharma collaborations, ongoing. Additional partnership deals would provide both platform validation and non-dilutive capital. Scientific conference data updates, ongoing, at venues including ASGCT, the European Atherosclerosis Society Congress, and the American Heart Association Scientific Sessions. Incremental preclinical and clinical updates, likely to be a recurring source of news flow between major clinical readouts. Section 16. Long-term outlook. Scribe's long-term outlook depends almost entirely on a single near-term question. Does STX 1150 work in humans the way it has in preclinical models? If the first half of 2027 data showed durable, safe PCSK9 suppression, Scribe would have genuine evidence that its ELXR epigenetic silencing platform can succeed in a large common disease population, a result that would likely accelerate interest in STX 1200 and STX 1400, attract additional pharma partnership interest, and support further capital raises on more favorable terms. If the data disappoint, either on safety or durability, the setback would likely affect market confidence in all 3 pipeline programs simultaneously, given their shared underlying technology platform. Over a longer 5 to 10-year horizon, the addressable opportunity in cardiometabolic disease is enormous, and a validated, infrequently dosed alternative to today's PCSK9 inhibitors could command a substantial share of a market currently worth billions of dollars annually. But that outcome remains several major clinical and regulatory milestones away, and the company will very likely require multiple additional financing rounds, equity, partnership, or grant-based, before any of its 3 programs could plausibly reach commercialization. Section 17. Bull case. STX 1150's first half of 2027 first-in-human data show a clean safety profile alongside durable, meaningful PCSK9 and LDL cholesterol reduction, validating the ELXR epigenetic silencing mechanism in humans for the first time. The positive readout accelerates investor and pharma interest in STX 1200 and STX 1400, both of which advance into the clinic roughly on schedule. Sanofi and/or Eli Lilly deepen their collaborations or pursue additional licensing and partnership deals, providing non-dilutive capital and validating the broader Cas-X and XE platform beyond cardiometabolic disease, and Scribe successfully raises additional capital on favorable terms as clinical proof points accumulate. In this scenario, Scribe could establish itself as a genuine next-generation leader in common disease gene editing, commanding a valuation well above its roughly $243 million IPO level as the market prices in a credible path to a large commercial opportunity. Section 18. Bear case. STX 1150's first half of 2027 data reveal either a safety signal or disappointing durability of PCSK9 suppression, undermining confidence in the ELXR platform broadly, and by extension in STX 1200 and STX 1400, which rely on closely related technology. Verve Therapeutics or another competitor reaches a comparable clinical milestone first, or already approved therapies like Leqvio continue to satisfy most patient and physician needs, reducing the commercial urgency for a novel and initially likely more expensive gene silencing alternative. Collaboration revenue remains lumpy and unpredictable, and the roughly $100 million of IPO proceeds proves insufficient to fund the pipeline through key value inflection points, forcing a dilutive down-round financing, and broader investor appetite for early-stage gene editing stocks, already muted since 2021, fails to recover meaningfully even as the 2026 biotech IPO market strengthens elsewhere. In this scenario, STCX shares could trade well below their IPO price as the market reprices the probability of clinical success sharply lower. Section 19. Final investment conclusion. Scribe Therapeutics enters the public markets with a genuinely differentiated scientific story, a Nobel laureate co-founder, a proprietary gene-editing enzyme distinct from the widely used Cas9, an epigenetic silencing technology aimed at delivering years-long benefit from a single treatment, and validation from two major pharmaceutical partners in Sanofi and Eli Lilly. That combination of scientific pedigree and commercial validation is not something every clinical-stage biotech IPO candidate can claim, and it helps explain why Scribe has been able to attract this level of partnership interest before generating any product revenue of its own. At the same time, investors need to be clear-eyed about where the company actually stands. STX 1150 has only just begun dosing its first human patients, with data still roughly a year away. STX 1200 and STX 1400 remain preclinical. Collaboration revenue is lumpy and unpredictable, and the company's own accumulated losses of $175.1 million through Q1 2026 underscore how much capital has already been consumed without a single approved or even late-stage clinical product to show for it. The roughly $100 million the IPO is expected to raise will help fund the next stretch of this journey, but will very likely not be the last capital raise the company needs before reaching commercialization, if it gets there at all. Bottom line, Scribe Therapeutics is best understood as a high-risk, high-potential reward, single catalyst story, ahead of pivotal STX 1150 data expected in the first half of 2027. Suitable primarily for investors comfortable with clinical-stage biotech risk and a multi-year time horizon. This report does not constitute a recommendation to buy, sell, or hold SCTX shares. Readers should review the complete Form S-1A on SEC EDGAR and consult a qualified financial advisor before making any investment decision.